Our first speaker is Ren Johnson, who has been studying some honeybees. Please welcome Ren.
Okay, hello everyone, I'm Ren Johnson, and the title of my project is What's the Buzz? Presence of a Honeybee Hive Decreases Native Bee Visitation to Native Plant Gardens in Northeast Ohio. So, just a question. What do you guys think of when you think of bees? Maybe stinging insects, oh, they're scary. Or maybe a lot of you think about specifically honeybees and something that we love, honey. But actually what a lot of people don't know is that there's a lot of other kinds of bees. Because honeybees are managed, we typically call them, livestock or something. But there's a lot of bees that we like to call wild bees, which are other species. You may know some, like the bumblebee, the sweat bee, mason bee, carpenter bee, or wasps. No, wasps are actually not bees, so they're actually not included in my research. While they're closely related, they're not the same thing. So, just the basis of my study, honeybees actually are not native to the Americas or Australia. You can see the areas in blue are where they're native to, so Africa, Europe, and Asia. So in the areas they're not native to, they're not necessarily used to the species in that area. So a lot of scientists have hypothesized that they could have a negative impact on our native bees in this area. So maybe, using the same resources, that can decrease the resources available to our native bees, in which case they would compete. So, why, and so in this case, oh, they're just another type of bee. Why do we think that these bees would outcompete our native bees? Well, actually, there's a lot of differences. So one thing is that honeybees are cooperative, and they do cooperative foraging. So, when you think of bees, you think of the queen and all the worker bees. So that's what honeybees do. They also do something called the waggle dance, in which case those worker bees can tell all of their other worker bees, oh, go to this giant patch of flowers. It's big. So they send all of their other worker bees there to really effectively make use of all of the flowers in that area. However, our native Ohio bees are mostly solitary. About 90% of our bees species in the world are solitary, so they don't form a queen. They really just raise their own young. And then there's some like bumblebees. Bumblebees also have a queen and they have worker bees, but they forage non-cooperatively. So they don't do the waggle dance. They're all just kind of doing their own thing. So this basically gives honeybees an advantage over other bees when it comes to finding resources. Additionally, there's honey. Honey is basically just a way to store nectar, which is what bees eat. So because honeybees make honey, they essentially can survive the winter, the colony can, and this gives them an advantage in the spring because it's like having, a pre-made army ready to go forage on all the flowers. Whereas this is not true for our native bees. So again, with the solitary bees, they overwinter as larvae, and then they emerge in the spring as adults and never get to meet their parents. And for bumblebees, the queen bumblebee also emerges in the spring, but she has to start her colony from scratch. So because of these reasons, basically that's why it's hypothesized that honeybees can outcompete our native species. So this leads me to my question. I was, wondering if this takes place on our college campus, specifically in our native plant garden that is located across the street from Ruth Williams Hall. So yeah, then here's my specific aim. This is a picture of the garden in August. I did this both in May and August, so you can see all the beautiful native species. So my specific aim is to determine the visitation rates of both honeybees and native bees to the plants in this garden. So the method that I used was calculating the bee visitation rate. So I did this with a honeybee hive, as pictured, present, and then also with it removed to compare. You can see me out in the garden. This is what it looked like in May. And so what I did is I basically threw a hula hoop. In case you're wondering, hula hoops are about 0.5 meters square of area. I threw that in there there for randomization, and then I watched that area for 5 minutes to count all of the bee visits, both honeybee and native bee. You can see a picture of a cute little bumblebee on a purple coneflower that I took a picture of. And so basically the bee landing on the flower was what I counted as a visitation. So then I repeated this on 10 areas per day for 10 days in May and 6 days in August of 2021. So now to get on to my results. So my results were significant. I got to see that when the honeybee hive was present, there was a significant decrease in native bee visitations than when it was absent. So that follows my hypothesis that, you know, the honeybees are a detriment to the native bees, basically by deterring them from visiting the garden. So then I broke it down more specifically by different types of bees. Because I counted honeybees, I did do honeybees, and then I grouped them into either size classes or taxa classes. So large bees, medium bees, Megachildae, which is a family of bees, and small bees. So as you can see, all of them trend towards there being less visitations when the hive was present. But only 2 of them showed significant results. Those were the honeybees and the small bees. The small bees, it makes sense because they were actually the most numerous type of bee in my study, as you can probably tell by this graph. And so basically because of that, I can kind of hypothesize that maybe if I had a larger sample size for some of these other groups, they may have also been significant. The one thing that's a little bit strange is that actually there were significantly less honeybee visitations when the honeybee hive was present. So basically, how I interpreted this was that when my hive wasn't present, the bees that were visiting the area were either from wild colonies or colonies kept by other people in the area. So then my hive being present must have deterred those bees also from visiting, but essentially because the number was so low, you would also have to hypothesize that my bees were not really visiting the garden. And I'll talk about what that means later. So what does this all mean in plain English? So 1, native bees visit the garden less when a honeybee hive is present. 2, the native bee's avoidance of the honeybee hive varies by the type of bee. 3, I did not observe many honeybees visiting the garden when my hive was present, so my bees weren't really visiting, and they were deterring bees from other colonies. So my theory basically is pheromones. So pheromones are chemicals that bees use for communication, and these are airborne. They can also stick to surfaces, things like that, and so it's like how they sense and communicate with other bees. So my hypothesis is that the pheromones from my honeybee hive, basically them being present, deterred both the native bees and honeybees from other colonies. Mostly because, the typical thought is, well, when the honeybees visit the garden, that's what's causing direct competition. But because I didn't see many of my honeybees visiting the garden, that can't really be the conclusion. So what did I learn from all this? Basically, don't place honeybee hives near native pollinator gardens. So first of all, there's no guarantee that they'll even visit the garden, just like with my bees. And then also, they could be scaring off our native bees whose populations are really declining, and we really need to protect it this time. So then, what did I learn from my independent study? Well, I learned a lot about experimental design. I made a lot of mistakes along the way that, now I realize I have a much higher appreciation for that. Additionally, I learned a lot of bee and plant identification. Found like over 400 bees or something that I had to identify. Lots of plants as well, so that's a good hard skill to learn. And then additionally, even before going into my IS project, I was really interested in insects and plants. And so this kind of cemented my interest in pollination research as a career. And then I just want to thank some people. So thanks to the College of Worcester for making this possible, the Henry J. Copeland Fund for Independent Study for funding my study, my advisors, Dr. Reed Johnson, Dr. Jennifer Eisen, and Dr. Hillary Edgington, and my second reader, Dr. Sharon Lynn, and then also my family and friends for supporting me along the way. Thank you.
For some question time, since we have a little bit extra while. Please share your questions for Ren.
So the honeybees were like bought from a like basically honeybee farm in California, but then they were shipped across the country. And when they were not at my site, they were taken to a research farm that's kind of closer to the OARDC.
What's known about like bees' preferences for specific gardens? I'm thinking like if they normally live near the OARDC, how is bringing them up to campus far enough for them to break any like preferences they may already have had for gardens down there?
Yeah, so it was far enough away that it would be very unlikely that they would forage there. So basically, bees typically forage within 2 kilometers of their hive. So that was far enough away that they wouldn't really journey all the way back to visit the site again.
So they would find new sites, and the thought was, well, why wouldn't they just go right next door with this garden?
Yeah, that's why we put them like in the garden. So when they didn't visit it, it was kind of like, what? You know what I mean?
Right.
Yeah.
That's why I was thinking that my other hypothesis was, oh, were they going back to their garden? Like, oh, fine, you can move me away from my garden, but I'll still go back. But no, it's too far.
Yeah, they like also like scout the area basically every single day, multiple times a day. So they're always looking for like new resources as well because flowers will bloom one day and then be dead the next day. Yep.
Since there is competition between the native bees and the honeybees, how can the lay person identify the difference so that they can kind of do a layout or plan to reduce that competition?
So I mean, like how you kind of tell the difference between them is typically just how they look. So honeybees look very distinct. Let me see if I have it. I think my first slide is a good picture. Yeah, so you see, like they all like look very different. So honeybees have that specific look. I almost say they're honey colored, but that's how you would tell the difference. But like how like a layperson would help like reduce competition is, like I said, don't put a honeybee hive in a garden full of native flowers.
So which of these do you really call, the wild bees? You would call them, no, which do you call the native bees?
All the ones on the right. So the bumblebees, the mason bees, the sweat bees, the carpenter
Those are the
bees.
native?
Yeah.
Okay,
And then the one on the left is the honeybee.
yep. Thank you.
Yep.
With the bee identification, was that all like photo based?
Yeah, so, well, one was by the time you look at so many bees, I can just look at a bee and kind of determine what, depending on what it is. So like bumblebees are pretty easy to tell from, a foot away or something. But a lot of them, if I couldn't identify what they were initially, I would capture them and then like take photos of them next to like a ruler. And I would submit those to either iNaturalist or I would look in a like bee book to see if I could determine what it was.
That's not your hand that bees on either.
No, these are all pictures I scrapped off the internet.
You mentioned that there might be some interesting effects from pheromones.
Mhm.
What type of follow-up study could you do to pursue that?
Yeah, so there's a lot of things. One is that we really don't know that much about native bees in general. So we know a lot about honeybee pheromones, but native bees, like what they use and what they can recognize, we don't know. So maybe like a full study using like a choice test, what are they attracted to, what are they not attracted to, just to identify those. And then potentially, additionally, is like to see for sure if it was the pheromones and not the foraging, is placing like, I know they have like those pheromone bags for like catching beetles and stuff, I'm sure you could do something similar using typical pheromones that are found around a honeybee hive and just placing that in the garden and seeing if that has the same effect as in this study.
So what advice would you have for our, local honeybee folks who are making their own, they've got their own hives? Is that essentially now going to be to the exclusion of our wild population? How do I think about that?
Yeah, so the cold hard truth is that there really shouldn't be honeybees in the United States at all. You know what I mean? Because like they are they're pretty bad for our native bees, and other studies have shown that as well. But that's not necessarily what's economically possible right now. I will say though that there have been multiple studies showing that honeybees are most certainly not the best pollinators of any given crop or anything. So there are different kinds of bees that you can buy. I know like mason bees and bumblebees online for like our native species and stuff like that. Really, the only kind of advantage that honeybees have is that they make honey. So, you know, you could specifically keep bees for the honey, but a lot of people keep bees for the pollination. And the truth is that there's always a native bee who does it better. You know what I mean? Oh, yeah.
Are the wasps an absolute enemy of a honeybee?
No. Well, wasps are basically like ecologically how they're more different is that wasps are more omnivorous, and people typically think of like most bees are like herbivores, so they eat like plant material.
Yeah.
And so I wouldn't say that wasps have never eaten a bee.
No,
You know what I
but
mean?
they're not by nature an absolute enemy of the
No,
honeybee.
no. They're just another, you know, insect that's in the environment.
One other question.
Mhm.
In your exposure to the bees and the honey and so forth, do you see hope for the beehive community rebuild?
You mean are people going to?
Well, they took such severe hits
Mhm.
for whatever. Do you see a comeback for them?
For the native bees?
Well, for the beehive bees to make honey.
Oh, you mean like honeybee numbers are decreasing as
Yeah.
well? Yeah. I think that there's definitely a possibility that that's a big area of research right now is how to stop the colony collapse
Okay.
disorder, which is what's causing the death
Okay.
of our honeybees. But I do think the preoccupation with like colony collapse disorder does detract from the reduction in our native bee species, you know, which are actually being hit even harder
Really?
than the honeybees. Yeah.
Really?
They're just much more vulnerable, and they don't have people kind of helping them like honeybees
First
do.
I've heard of that. Thank you.
Oh, yep.
In your study, did you identify where the native beehives were?
Yeah, so I did not do that mostly because that is incredibly hard to do, and a lot of, again, we know very little about our native bee species. A lot of native bee species, we don't even know what their nests look like because no one has ever found a nest of them. Like, that's how little research there is on them. So, no, I would have, like, any kind of, like, tracking device that you could put on them, the bees are just too small. And the only alternative would be chasing them and hoping that I see where they land. So, no, I didn't.
Let's thank Ryan one more time. It's really nice to have such a lively Q&A. Thank you, everyone. Our next speaker is Nathan, who will be taking us over to some plants. Please welcome Nathan.
Alright. Hi everyone, I'm Nathan, and I'm very excited to share with you my. Okay, I used to okay. I'm very excited to share with you my independence today. It's about this fascinating and increasingly prevalent interaction between soybean and the soybean aphid. So with this interaction, let's just start by looking at each of the characters, if you will, that are involved. Starting with the plants. So soybean is a species of legume from East Asia, and it's one of the world's most important crops. And Part of that is because it's a rich source of high quality protein for human food and animal feed, as well as being a major source for vegetable oil. And then the insect in the interaction, similarly to the plant, it's also natively from East Asia and it is capable of reducing the soybean yield by up to 40%. It was initially discovered in the U.S. In around the year 2000, so it's a relatively, on a biological scale, a relatively recent event. And because of all this, it is a significant agricultural pest. So, why is this interaction significant on a broader scale? The global population is rising, and generally, when you have more people, you need more food as well. But invasive species like the soybean aphid limits the important agricultural yield, and the U.S. is expected to continue accumulating invasive species. And to no surprise, really, it will all come down to money eventually. More agricultural pests means more money is required to manage them. So how do we manage them then? There's a few ways. Primarily, chemical management is being used, like through the use of insecticides. However, using insecticides can get expensive, and it is also harmful to the natural enemies of the soybean aphid. And this actually already happened now. The soybean aphid has developed resistance to insecticide. But there's also host plant resistance. And host plant resistance is environmentally and economically more beneficial. And I'm sorry, that's a mess out there. Okay. And host plant resistance is conferred through the expression of naturally occurring resistance. And that's part of the reason why it's a lot less expensive to use, because they're naturally occurring traits. And in the soybean specifically, they are expressing RAG genes. And RAG genes it stands for resistance to aphis glycine. Aphis glycine being the scientific name for the soybean aphid. So when plants detect wounding, they mount various forms of defense, and the effectiveness of RAG genes has been under pressure. So soybean aphid, the effectiveness of this naturally occurring resistance has been under pressure because of biotypes of aphids being discovered. And biotype is just a label that kind of describes if the aphid is able to overcome specific RAG genes. So there are multiple aphid biotypes, well, 1 to 4, but most relative to this study, biotype 1 and biotype 4 aphids. As you can see by the table, so a biotype 1 aphid is able to survive on susceptible soybean not having any of those naturally occurring RAG genes that can combat the aphid. However, they cannot survive on the resistant soybean because they have the resistant soybean has these specific RAG genes that can suppress the soybean aphid's colonization. And then, most interestingly, and I believe the biotype 4 aphid was discovered around the year 2013, so again, even more recently, the biotype 4 aphid is able to colonize susceptible plants like the biotype 1 aphid, but is also able to colonize the resistant soybean, which has those RAG genes. So the discovery of specifically the virulent biotype 4 aphid has focused our attention on understanding how the mechanism of RAG resistance works in the plant. Specifically, how does the phytohormonal response function? So phytohormones are similar to hormones in us, except they're specific to the plant. So phytohormones are signaling molecules that mediate defensive signaling pathways, which utilize the RAG response. And there are multiple key hormones involved in the reg response, but we'll be focusing on just one, and that's desmotic acid in this case, which I'll probably start referring to as JA. But in this study, I analyzed the phytohormonal response during soybean to soybean aphid interactions, the expression of phytohormone responsive genes, specifically a JA responsive gene. So the goals of the study, there are two, and they're pretty interconnected. First, understand the patterns of soybean defense signaling during interactions with biotype 1 and biotype 4 soybean aphids. And then second, and more specifically, establish a refined understanding of phytohormonal changes during interactions with biotype 1 and biotype 4 aphids. So this is how I did that. These are my methods. So I have biotype 1 and biotype 4 aphids, and I'm manually infesting those onto either a susceptible plant or a resistant plant. And then after either 3 hours or 9 hours, I remove the aphids and I collect the soybean leaf. And then with that soybean leaf, I extract the RNA and I perform RT-qPCR. So I just have the goal there as a reminder. So we're trying to establish a refined understanding of these phytohormonal changes. And the RT-qPCR results primarily reveal there is a change in the gene expression of this phytohormonal responsive gene. However, some of the results were unexpected. Okay, so this is kind of a lot to look at. So I put up a few different colored stars there for reference, but we'll refer to those later. But this figure is showing you the 2 time points. You have the 3 hour and the 9 hour. And within each time point, we have our susceptible plants and our resistant plants. And the color of the bars refer to if the plant was uninfested, or if it had biotype 1 aphids on it, or if it had biotype 4 aphids on it. And then the height of the bar on the y-axis is the relative fold gene expression. And then those little letters above the bars that you're seeing indicates a lack of statistical significance. So if bars share the same letter in any way, they're not statistically significant from each other. So there's a lot of variation, as you can see, because it's a little strange. It's a little strange to look at the, for example, the susceptible 3 hours, and the bars all look kind of different from each other, but based on the letters, they're all not significantly different from each other. But since finishing this portion of my IS and increasing the rep number, the variation has increased. The gene expression is similar to the uninfested control in the susceptible plants after 3 hours and 9 hours. And this is expected because both of the aphid biotypes are able to infest the susceptible soil. So you can think about it where a lower gene expression generally would mean less phytohormonal activity that is regulating an effective defense response. However, if you look at the blue stars, which are the biotype 1 aphids unresistant, it would have been expected to see a higher gene expression since the biotype 1 aphids cannot infest the resistant soybeans, but we're seeing the opposite of that. And this may be due to there being a different phytohormone in play, which was shown in previous studies to play an important role. That just wasn't analyzed in my study. And then, probably the most interesting, if you look at the red star, which is the biotype 4 aphids on resistant soybean, it was unexpected to see the significant increase in gene expression since biotype 4 aphids can infest the resistant soybean. So for this interaction in particular, we can ask, why are we seeing a seemingly higher defensive response in an interaction where aphids can overcome the defensive response? So to answer this, we need to understand the basics of the jasmonic acid pathway. So VSPB is the gene, the JA responsive gene I was looking at. VSPB is more specifically responsive to methyljasminate. Methyljasminate within this jasmonic acid pathway is a downstream conjugate of jasmonic acid, and it is also metabolite of the further downstream jasmonic acid isoleucine. And the JA isoleucine is the bioactive form of the phytohormone, meaning that JA conjugate is what's triggering the expression of RAGE genes. So methyljasminate is kind of generally sitting in between the bioactive form and one of the more initial forms of jasmonic acid. So knowing this, then, a low expression of our VSPB gene that we looked at in interactions when aphids are able to colonize susceptible soybean, like we saw, which was expected. This may be due to a blocking or some kind of evading mechanism of the JA pathway that happens before the methyljasminate. So this would then explain that low expression of the gene expression values for that JA responsive gene. But then to address that high amount of methyljasminate expression in biotype 4 is on the resistant soybean. That may be due to a blocking mechanism before the JA isoleucine. This is possible because previous research has shown a decrease in jasmonic acid isoleucine in the same reaction, while we're seeing an increase in the metabolite before jasmonic acid isoleucine. So, considering this, the major results suggest that the involvement of the jasmonic acid pathway varies depending on the soybean aphid interaction. Biotype 1 or biotype 4 susceptible and biotype 1 resistant interactions are compatible, but they involve different plants, different biotypes, and result in different phytohormone expression levels. Understanding the variation in phytohormonal responses can reveal the nature of incompatible or compatible interactions between soybean and soybean aphids, and aid in future research involved in determining the source of virulence from the aphid's perspective. And these patterns can also be useful for guiding future research involved in enhancing the durability of RAGE genes and understanding the important phytohormonal aspects of the RAGE response. Thank you.
Let's hear some questions for Nathan.
I didn't get your last name.
Oh, Kreider.
So when you were showing your graphs, were you only testing for the middle part of the pathway?
Yeah.
What do you think you would have learned more if you guys tested for both the beginning of the pathway and the end of the pathway?
Yes, so I'll just go back to this quick. So if we tested for the end of the pathway, the jasmonic acid isoleucine, we would have expected, let's go back to, there we go. So if we're looking at that red star there, in that same interaction, if we are testing for jasmonic acid isoleucine, we'd expect that bar to be much lower. And then in the other interactions, like with the blue star, we would expect to see a higher expression of a conjugate of jasmonic acid that happens before that. Yeah. Or actually, we might not even see anything, because there's also a question of, is the plant actually detecting the soybean aphid? And if it's not, then it might not be a change. But if it would be lower than the control, then I think it would be more of a blocking. So it's kind of a weird distinction. So I just put it together, blocking or inhibiting for right now. Yeah.
I might have missed this in your presentation, but why did you choose jasmonic acid?
Oh yeah. Jasmonic acid is it's generally involved in a defensive response in plants. And previous research has shown that Specifically with reg genes, there is some kind of role that desmonic acid plays that is involved in activating those defensive responses. So it's just a defense regulating phytohormone.
You mentioned that you were thinking about replicates on this slide. How many replicates do you have here, and what do you think happens when you have more replicates that you mentioned?
Yeah, so here we have two replicates. Well, two of. So, like, if I was putting biotype 1 on susceptible, I'm putting 15 aphids onto one leaf, and then I do that 5 times, and then that's one replicate. So I did that twice here, and I have a third replicate that I've still been looking at, and what I'm seeing from that is a decrease in those error bars and just a reinforcement of these trends that we're seeing.
How do you differentiate between a susceptible and a resistant plant? Like, is a resistant plant, does it have the jasmonic acid? Susceptible plant does not.
So susceptible and resistant plants both have jasmonic acid as a phytohormone, but what's different is susceptible does not have these specific reg genes against the soybean aphid, while the resistant plant does. And those, the presence of those genes in the resistant plants are activated by jasmonic acid. So it's a difference of specific genes. Yeah.
Do you think your results would have changed at all if you had observed this past 9 hours? Would anything have changed?
Yeah. Probably. Yeah. So other studies have done it within 24 hours. So and then even past 24 hours, like on a monthly scale. I specifically looked at an early time point because the early time point is a very important stage within an infestation period, because if the plant can't deal with it early, and the aphids are able to stay on for a long time, the plant has to constantly keep using energy to mount these defenses throughout its growing stage. So if I used a longer time scale, maybe with the I think previous research has shown that the jasmonic acid response has typically decreased, and that's probably a part of them being able to deal with the aphids, or I mean with other phytohormones, the aphids increase the expression of a different phytohormone that represses that one. There's many phytohormones involved. This is just one of them, and they have very complicated interactions between each other. So it's kind of complicated.
What is the result of this response for the aphids? Does it lead to the plant protecting itself by killing the aphids, or how does it protect itself? And what is the consequence of your study for soybean crops in general?
So what's happening directly to the aphid when the plant mounts a response? So when these reg genes are expressed, there's a variety of things that happen. One of them is a change to the cell itself, the plant cell, and that would be an increase in thickness of the cell wall. Because when the aphids feed, they have this needle mouth part that kind of probes around on the leaf surface, and it sticks into the plant, trying to find the sugars of the plant to feed on, and increasing the cell wall thickness would make that harder to do. And then plants also produce these things called secondary metabolites, and when those are ingested by the aphid, it's toxic to it, and it can either deter it or kill it. And then your second question, can you repeat that?
What was the consequence for soybean crops? These findings, how do they impact soybean farmers, for instance?
Yeah, how do these findings impact current soybean crops and farmers? So right now, a lot of research is trying to understand how the rate mechanism works and how soybean aphids are able to overcome that. But right now, all the soybeans we see outside are basically susceptible. Reg genes are being studied from these past soybean lines that are frozen in seed banks. And they haven't really been incorporated into the farms outside we see today because insecticides have been the primary use. And ever since recently, the insecticide resistance has been discovered in the aphid, the major soybean seed lines or companies are saying, okay, so what were you saying about Reg genes? You know? So, because it's getting expensive, so they don't like spending money, obviously. So right now, it's more, a lot of the research is just understanding what is the reg response, because there's multiple reg genes, like what does this reg gene do, what does this reg gene do, which one should we include in a mass-produced seed line. So it's kind of up and coming right now, I would say.
Did you run into any roadblocks while conducting your study, and if so, how did you overcome those?
Yeah. Give me flashbacks. So, I mean, there's roadblocks at almost like every step of the way. Some are probably easy to overcome, obviously. Some of them, like, sometimes the seeds wouldn't grow. It's like, okay, let's replant, I guess. Performing the RT-qPCR was a big learning curve for me too. So at the start of that, the values I was getting from doing RT-qPCR were inconsistent, and generally when you do RT-qPCR, your numbers need to be really tight together, and mine weren't. So I had to kind of just run plates that had changes to them, so I was just troubleshooting it, you know, and let's see, those are the primary ones. I mean, I had a different study that I was working on that just didn't work, and it just didn't make it to my IS, just because the gene target wasn't getting good results. It just wasn't a good gene target to look at. So that was just something that was completely ditched. So, yeah.
Very specific question.
Yeah.
I'm interested in pesticides in my own research. Do you happen to know what are the most common insecticides used?
No, I don't.
Okay.
No,
Going back to the slide, there is a difference between the 3 hours and the 9-hour period, and you might have mentioned this already, but what is the significance of those two time periods? That's my first question. And how do you interpret the difference in biotype 4, and why there's not a significant difference in 3 hours and there is no?
So, yeah, so the significance between the 3 hours and 9 hours was just to see the change over time, really, because previous studies looked at just one, like a 6 hour, and so I just wanted to do 3 hours and 9 hours. Another roadblock, I tried to do 6 hours, but I didn't have time to do that. So I chose the two on either side of that, 3 hours and 9 hours, to see if there was a significant increase or any kind of change. And maybe doing a later time point would make it more clear, would make those trends over time more clear. But that didn't turn out to be a major result in this study. I mean, we are seeing, you know, if you look at 3 hours and 9 hours from the Biotype 4 on resistant plants, it's changing. That gene expression is increasing over time from 3 hours to 9 hours. So that's giving us some sort of information about the behavior of can you repeat your second question? Oh, I did, okay.
Any final questions for Nathan? Let's thank Nathan. This concludes the session on ecology and the environment. Thank you for being a wonderful audience. There'll be a short intermission, and the session on modeling applications will begin at noon.
It's the owl. What's your impression of the recording? So I'm kind of one, are you yes. So your poster session.
I'm Lindsay, yeah. Nice to meet you. What was that? The baby? Yeah. Well, nice to meet you. I'm just you know. Good.